This set of notes is going to be all about DNA, how it gets replicated, and replication of DNA means copying it. So taking one DNA molecule and making two, which is a very critical part of life, because to make one cell turn into two cells, we have to copy our genetic material, which is the instructions for making protein and doing all of the stuff that life does. So this is going to be part historical, looking at some scientific experiments that have been done to give us the information that we have, and then just part technical. What's going on as far as the best that we understand it today. We're going to go back starting about 100 years ago at first, and then we're going to see that a lot of amazing biotechnology has happened in that time frame, giving us some really cool techniques and capabilities today, and things that you guys need to know about, not just as biologists and scientists, but as just scientifically literate people, so that you can decide things when you come to voting and figuring out as a society, what are we okay with doing, and what sorts of things are we going to say is too much? And you need to really understand what that technology is. So in 1928, a scientist named Griffith took and made an experiment out of some really simple means. So he had two different types of bacteria. These types of bacteria were called, for the experiment, S cells, which these ones were pathogenic, meaning disease-causing bacteria. You take these types of cells, you give them into an animal, and they cause sickness. There were also some related bacteria. There were R cells, but these ones were non-pathogenic. They did not possess whatever it took to spread and grow inside organisms, and they were non-sickness causing, similar to lots of bacteria that are around us that don't cause huge, terrible diseases. What Griffith did, aside from these two control groups, was to destroy the S cells using heat. And in that case, just like when we cook food, we are heating up proteins to the point where enzymes denature, change shape slightly, and they don't work. And if you damage enough critical enzymes that are important for doing the jobs that they do, you have, in essence, killed the organism. And when we cook our food, we go ahead and eat all the dead bacteria there without any ill effect. But what Griffith did was he took some heat-killed S cells and mixed them with some living R cells. Now, just mixing them was all that he did. He didn't introduce any other sorts of biotechnology here. And something occurred there that caused the mouse to die, so that those R cells that were non-pathogenic became pathogenic S cells. So something about those dead or heat-killed S cells was still useful and still what we call transformed into those R cells. And anytime we take a gene or a unit of genetic code from one organism and put it into another organism, we call that process transformation. And we call that new piece of DNA that now incorporates two different pieces into it, recombinant DNA. It has been recombined with another piece. So another experiment, this is a little bit later, in the 50s. So this was done by a couple of scientists named Hershey and Chase. And what they were interested in, they were studying bacteriophages, which are viruses that infect bacteria. Bacteria are susceptible to viruses just like you and I are. As a matter of fact, there are a lot more viruses that infect bacteria than there are viruses that infect the rest of the world. So what they were interested in is there were, at the time, there was currently a big scientific debate about what it was that passed down the genetic code from one organism to the next. We knew about genes, we knew about Mendel, had done research with peas. People were starting to really hone in on what molecule is responsible. And it was known that there are some things like DNA and RNA floating around, and we also knew that there were proteins involved. And people have been studying proteins for a few decades. And it was known that there were millions of different types of proteins, and the proteins had uh, a code within them that was spelled out by what is it that makes a protein. So proteins are spelled out by amino acids, and there were 20 different amino acids that we found being used. Whereas in DNA and RNA, we had 4 or 5 nucleotides that were used, just different nitrogenous bases that were associated with them. So it was thought at the time that DNA is something that's extra in the cell. There is no way that you can spell very much useful stuff with just 4 or 5 letters. Whereas if you had a 20-letter alphabet, as we do with proteins, then you can make a lot more unique and different stuff. So Hershey and Chase set out to discern, okay, is it the DNA or is it the protein? And so they took some phages. These are the bacterial viruses, right? These are phages. So they took the phages and they grew them in different environments. One of them, this batch one, they grew in radioactive sulfur. You will recall that sulfur is a component in one of our amino acids. It's important for making disulfide bridges. So it is going to be found in protein. The other batch they made with radioactive phosphorus. Phosphorus, as you know, is a component of the nucleotides throughout DNA. They have a sugar, a phosphate, and a nitrogenous base. So as they grew these, the DNA would have been labeled with radioactive phosphorus, the sulfur would have been grown, and all of the proteins would have incorporated radioactive sulfur into them. So then they're able to do this experiment. So they let their labeled phages, and either labeled with radioactive protein or radioactive DNA, infect some cells, and then they put them in a blender, and then they centrifuge them, and they separated out the DNA and the protein. The DNA was more dense, and the protein was less dense. And what they were able to find was, sorry, not the DNA, the cells, the cellular mixture that had settled to the bottom, and the rest of the viral particles left at the top. What they were able to find is that when they did this first experiment, the radioactive part remained up here at the top, what's called the supernatant, or in the liquid, and that was not incorporated into any of the cells. The pellet was not radioactive. What they found in the batch 2 was that some of the radioactivity, that the radioactive part of these had been injected into the cells and was now in the cells. So this was a confirmation that DNA is important for passing down genetic information. At least in the context of viruses, they injected DNA, or sometimes that we know they inject RNA into foreign cells. So we know that nuclear information is done on what we call DNA and RNA. So now that that was done, we can start to associate a couple of other things with it. So right around the same time, just a couple years prior, there was a researcher named Erwin Chargaff, and Chargaff did some, in class normally we'd read the research paper, but basically he came up with, he studied a wide variety of organisms, he isolated their nitrogenous bases, and he found that the DNA, the amount of DNA is not always the same, and the amount of adenine, thymine, guanine, and cytosine are not always the same. But he did happen to make a note, as a really astute scientist he was, that adenine and thymine were always roughly equal. And in his results, they weren't exactly equal. They were close to equal. And guanine and cytosine were also close to equal. Now, he didn't actually know what that meant at the time because nobody had seen DNA. Still, nobody has really seen DNA. But he did note that it was significant, and he did publish the results. And some other scientists were able to put that together and come up with the double helix model. So 1953 was done by a couple of chemists, James Watson and Francis Crick, and they were able to propose the first model for DNA. Again, we would read their paper normally in class. It's just a one-page paper that lays out the structure of DNA, and they're just dropping a bomb on scientific community. And the level of chemistry knowledge that these gentlemen had is pretty intense. I'm going to show you the information that they were able to use to put this together. But they were able to give us this model, and they were able to make some assumptions and guesses just in that one-page paper that it automatically did make sense that DNA would be the molecule that is used to get copied and pass on information to multiple generations. So here's sometimes what we end up thinking of in terms of what DNA looks like. This is a ribbon model. What it actually looks like, we can't see it. The closest electron microscopy gives us a really, really fuzzy picture of it. So this is the model that Watson and Crick proposed in the 1950s. And the model that they were able to build and make, and this shows the phosphate backbone in a double helix, and they've got some clamps holding some of the purine and pyrimidine bases, nitrogenous bases here on the inside. They were able to do this using Chargaff's rules, that adenine and thymine were always roughly equal, and guanine and cytosine were also always equal. They were able to do this knowing the composition of DNA, meaning that it was deoxyribose, phosphate, and these nitrogenous bases. And this image, which was provided to them by a colleague, Rosalind Franklin. So this image is X-ray crystallography. It's not an actual photograph. It's a photograph of a crystal structure of DNA that's being hit with X-rays. So how you take this and get to this structure, and furthermore this, requires a pretty healthy dose of physics. So Franklin was the one who actually did the X-ray crystallography. Crystallography is still a thing. It's done not just with nucleic acids, but also with proteins, and proteins is a big area for it currently. It's as much art as it is science. Franklin was the one who was really good at this technique, and she actually did some presentations and proposed that it was DNA. Currently, at the time, people thought that the phosphates were on the inside and the nitrogenous bases were on the outside. She was the one who posited that the bases were on the inside and the phosphates were on the outside. But she didn't have the chemical background and the physics background quite to come up with the level of explanation that Watson and Crick were. And she's a woman, so she didn't get very much credit at the time. She was mentioned and cited in their paper, but they were the ones who got all the glory. So. All right, so we're gonna look at a few different models of DNA and RNA structure and a couple of different video clips. These are going to advance in their technical nature as we go on throughout these. Here we go.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these three bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group.
In organic chemistry, they always number carbons and carbon chains. They start with the carbon that's bound to the biggest molecule. In this case, the nitrogenous base is the biggest thing that is bound to it. So the number one could either be here or here. But in this case, number one is bound to the nitrogenous base because it's largest, and then it just goes 1', 2', 3', 4', and 5'. Those are going to become important later.
The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
So we're going to see that this business of nucleotides always being built in the 5' to 3' direction, or new nucleotides are only capable of being added to the 3' end that is hanging off, as being both an evolutionary relic and a problem that has to be dealt with by organisms in terms of DNA replication.
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
And genes can be just a couple hundred or several thousand nucleotides long. So we saw our DNA was set up like this, with the labels on these are 5' end and 3' end, just because those are the places that happen to be hanging off. On the 5' end, there's a phosphate group. On the 3' end, there's a hydroxyl. We're not going to stress too much more about what goes on, but in the rest of the structure, except that it does have a directional nature to it. And you did see that those two strands are anti-parallel, so they're oriented backwards, and DNA can only be built in the 5' to 3' direction. So you already know that a nucleotide's got three parts. It was on your last quiz. Here's a space-filling model showing some of another way of illustrating DNA. So inside DNA, if we open it up and take a look at it, it is theorized, and was theorized by Watson and Crick like 70 years ago, that guanine and cytosine could potentially pair up because there were places within them, or on their ends of those nitrogenous bases, where hydrogen bonding could happen. And between guanine and cytosine, there are 3 hydrogen bonds. Between adenine and thymine, there are 2 hydrogen bonds. Do take a note that we always link up a purine, one of these big molecules, with a pyrimidine, one of these small molecules. Otherwise, the spacing between the two backbone pieces would not be uniform. So we always have the same space between them. 5', 3', 3', 5'. I think that's all we need to worry about on there. And you can see on both of our model structures that are spaced out that DNA is not exactly just a spiral with a second spiral with it. It is a double helix, meaning that we've got a space between these here, called the minor groove, and the space up here called the major groove. So they're not exactly they're not spirals that are evenly spaced. And the rest of the data about this we're not going to worry about. So this shows you that we always have to have a purine and a pyrimidine to give us consistent width. And Watson and Crick were able to explain why Chargaff's rules worked out, because adenine had to bind with thymine, so therefore adenine and thymine must be the same percentages. So if adenine accounts for 20% of an organism's DNA, if 20% of the nitrogen spaces within DNA, then thymine would be about 20%. What's 20 plus 20? We'd have 40. That means the remaining 60% is accounted for guanine and cytosine, roughly 30% each. Hopefully that makes sense. So which one of these is harder to separate, the one with 2 hydrogen bonds or the 3 hydrogen bonds? That's actually relevant because for genes to get access, we've got 2 strands of DNA, and they don't get just magically translated into protein. The DNA has to become opened up, and it has to, whichever strand has the gene on it, has to get read by some machinery that we'll see in our next unit. Well, it turns out that right at close to the beginning of genes, there are usually a whole bunch of A's, or adenines, and thymines. They call it there's a sequence that is prior to the gene starting that has a little bit of an easier region to open, and those multiple adenine and thymines right in there help out with pulling apart that DNA. Watson and Crick also proposed that the fact that the DNA was double-stranded and that each side could essentially be used as a template to build the other side proposed a copying mechanism. And it does make sense that if one side has to match up particularly with another side, that there could be some copying. So there are 3 different models that could be proposed for how DNA gets copied. One, where we take our DNA, we make an exact copy of that DNA, and then that exact copy of DNA gets copies made of it. 2, where we take that DNA, split it up, and then end up making a copy of each piece, and then as that splits up again, we would make a copy of each piece. And then a third model that DNA could be copied in some sort of a dispersive fashion, where we have individual pieces of DNA going to different locations and gradually dispersing the old strand with new strands. So in these models, the pictures, to help you out here, the original DNA is shown dark and the new DNA is shown light. Go figure, this relates to an experiment. So this experiment involved taking some heavy or dark DNA that was cultured for a long period of time in heavy nitrogen, so nitrogen with an atomic mass of 15 instead of 14. Nitrogenous bases are all going to be built with this nitrogen. It's all going to be heavy. Then they take the bacteria and they allow it to grow in a medium with regular nitrogen in it. So that old DNA that's in these bacteria is still going to be there. The question is, how is it going to be there? They took some DNA out of this after one round of replication, after say 30 minutes to an hour after the bacteria had replicated, and found that that DNA was all uniform density. They pulled out some after another 30 minutes to an hour, after it had replicated one more time, and they found that a large quantity of it was still very dense, but they also had a second band that was less dense. So what model does this information match up with that would give us that same type of information? So it turns out that we do have semi-conservative copying of DNA. We have from our parent strand, it does split apart, and each one of these strands is used as a template for new DNA to get built. So the formation of these, we'll see, is kind of complicated. So DNA gets copied pretty quickly. If you know anybody who's a fast typist, my guess is that they cannot do 500 keystrokes per second. They probably can't even do 50 per second. So bacteria and prokaryotes can do a little bit faster than us, but still, it's lightning speed either way. There are errors that occur because these machines are not perfect, and occasionally some environmental factor can interfere with the machinery, and we think that there may be some random error built in the machinery anyway. We usually use bacteria to model this out, so we're going to see E. coli is mentioned a lot of the time. E. coli is a model organism. It lives inside us. It's really easy to study in a petri dish, and it's relatively simple, we think, relative to what goes on in eukaryotes. However, the processes are mostly the same. So let's take a look at a couple of videos. They're going to get more advanced as we go. See if this is
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles, which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Hopefully that makes sense. It's a start. We're going to see some of those terms like polymerase. It's an enzyme that makes DNA polymers. So hopefully, I guess that makes sense. So origins of replication is just specific regions where these proteins bind, open up or unzip the DNA and start replicating it. In eukaryotes, we have many origins of replication. Our DNA polymerase doesn't work quite as fast. So it does make sense that if we've got a big long chromosome, that multiple locations would open up and replicate. In prokaryotes, they replicate DNA something like 10-fold faster. They only have one origin of replication, and they unzip and open up and replicate their entire chromosome that way. You're not going to need to panic about those names this year. So in E. coli, which is a prokaryote, we can see that it has one origin of replication. It opens up. Because it is circular DNA, remember, this is still a double helix. It is in a complete loop. So we've got polymerase molecules extending in both directions. We've got one strand that is continuous and the other strand that looked like it was done in pieces, because DNA can only be built from 5' to 3', or they can only be added in the 5' to 3' direction. In eukaryotes, we have multiple replication bubbles that happen all at the same time. So how does DNA replication start, aside from DNA gets opened up into a replication bubble? Well, it turns out that that our DNA polymerase has a technical problem. It can only add nucleotides. It can't just like start a piece of DNA. It can only add nucleotides to an already existing chain, which is a problem because we need to make a brand new strand. DNA polymerase is able to do this by waiting until there is a little piece of RNA that happens to match the base sequence of the piece of DNA that's getting copied. This piece is called a primer. Then RNA happens to stick on there. DNA polymerase grabs on to that primer and then adds stuff to that. So eventually there will be a DNA polymerase that comes along and replaces the original piece of RNA. But we do get all our DNA gets started with a little section of RNA, and we're talking just a few nucleotides long. So the new DNA strand will always grab onto the 3' end of the RNA primer. We said that DNA polymerase can only add stuff to an existing 3' end, to a hydroxyl end. So that's an evolutionary problem and solution, but it's something that exists in every organism on Earth. As these monomers are added, it loses some phosphate groups. So DNA polymerase is an enzyme. How does that enzyme work? Well, it doesn't provide the energy. It just uses, it allows a reaction to take place. The reaction that takes place is that the nucleotides that are ready to get added onto pieces of DNA have extra phosphate groups on them that provide the energy. So DNA, we say it's built in the 5' to 3' direction. The 3' end is always the new piece, and we're going to add a new piece on beyond that 3' end. So this is just showing us what a nucleotide looks like before being added to a piece of DNA or RNA. So this right here has some extra phosphate groups. It has extra potential energy, if you will. And then polymerase inserts this nucleotide, and then as a nucleotide, as we usually think about it in DNA or RNA, it's just one phosphate group that's left. So here's showing a replication bubble. We've got several enzymes at work here. We have an enzyme that is responsible for opening up the double helix, so breaking those hydrogen bonds apart. And as we go down here, we're going to have a DNA strand getting built in the 5' to 3' direction. We can't have this strand getting built at the same time in the same direction because these pieces are anti-parallel, right? So this other strand is built, again, in the 5' to 3' direction, but it has to go in the opposite direction, which means as this DNA unzips, it's going to have one side that's made continuously, and it pretty much paired up immediately as it is unzipped. It's going to have another side that's left wide open for a large period of time, relatively. And that introduces a big possibility for mutations and damage to that DNA. When there's 2 strands of DNA, if there's damage to one strand, there are enzymes that can come and read the backup and make whatever repair needs to be made. If there's just a single strand of information and it gets damaged by X-rays, gamma rays, anything like that, that's it, oftentimes, and there's no backup to repair it. This is why radiation tends to take out your fast-growing tissues, your hair, your blood, or skin. That's some of the first things you notice with radiation exposure and radiation poisoning. So here's what we got going on. Primase attaches an RNA primer to a particular region that happens to match up to this RNA primer. If RNA primers were hundreds of bases long, there wouldn't be any places where these primers would happen to exactly match up. However, since they're only 5 to 10 nucleotides long, there are matching sequences all around in our DNA. So from this, we're going to have DNA get built, attaches onto the 3' end and builds new all the way down here. It will eventually replace this other primer, and then we're left with a fragment to get repaired. There's another polymerase that will come and kick off these primers, and then there's a ligase enzyme that will DNA polymerase 1 comes and sticks these guys down. They're named in the order they were discovered. You don't need to worry about it. But comes around and deletes this RNA, and then there's an enzyme called ligase. Ligase, like ligature, like ligand, it sticks things together, and it makes what we call a phosphodiester bond between the phosphate group of one molecule of DNA and the 3' hydroxyl group of the other one. So these discontinuous pieces of DNA are called Okazaki fragments. Can you guess what country the person was from, the scientist was from who discovered them? Japan. All right. So they're made in pieces. This is called the lagging strand, and it is a place where damage can occur if it's being exposed. All right. So we're going to watch our BioFlix version of this, and we're also going to watch a way more advanced, better version of this video. Let me see if I can pull these up. I lied. I don't have my BioFlix one ready. We're going to skip straight to the advanced one. Enjoy.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom.
Which one of those strands is the lagging strand from the picture on here?
The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Thanks for leaving out the extra details so we didn't get completely confused. I appreciate that, guys. So, yeah, there's a lot going on, and we just know pieces of it so far. There are new proteins and enzymes that are learned about every couple of years with this. So I think you guys have seen enough of that stuff. DNA not only gets built super fast, it also gets proofread by DNA polymerases. So they have multiple jobs here, and any nucleotides that get paired incorrectly, they're not going to fit quite right. An enzyme is a shape-driven machine, and it comes along, finds the places where the shapes are not right, deletes and replaces nucleotides that don't work. And this happens in the ballpark of, a few hundred nucleotides per second. Remember, organisms like us have something like 1,400 biology textbooks worth of DNA within us, so it has to be done pretty quickly. Mismatch repair is one type of repair. So this is where we've got like adenine, instead of being bound with thymine, might be bound with guanine or cytosine instead. That's one type of repair that's being done by a type of a polymerase. And there are colon cancers that are like this. So I actually have to go and get a colonoscopy next year because my dad last year had colon cancer, and they're saying that that I might have inherited a bad gene for this mismatch repair. So that's fun stuff. You guys probably are not aware of how much your DNA is damaged on a regular basis. We're exposed to carcinogens all the time. Remember, oxygen that you're breathing in and using as respiratory potential is slowly killing you. It is a powerful oxidizer, probably the biggest carcinogen in most people's regular old environments. And the repair mechanisms by these are usually quite seamless. So there are nucleases that are enzymes that cut out damaged sections and then put in correct ones. So this child right here has a disorder by which she cannot damage or cannot repair thymine dimers. That's this right here. I get a couple of thymines. UV damage. The number one UV damage is to make these little guys, and it's a really easy repair to make for those of us who have the enzymes to make that repair. You all have experienced far more sun damage than this child has had. It's just that your DNA has all been repaired, and she's going to die at a really young age from melanoma. So the proofreading machinery that is there taking care of all of our damaged DNA is very significant. And when we inherit bad proofreading or bad repair technology, that's when we have higher incidence of things like cancers coming after us. So after proofreading, we talk about our error rate normally being 1 in 10 to the 5th, being reduced roughly, we're down to like 1 in trillions of nucleotides. So that being said, we do still have more than that in terms of our DNA. So you are going to have some mutations within your DNA as this gets copied. And the sequence changes, if they are located in the germline, in your germline, where are your germline cells located if they're going to be inherited? If there are sequence changes in your germline, which would be in like your testes or ovaries, in the cells that will eventually become eggs and sperm, those can be inherited. And is that going to be a good or a bad thing for your next offspring? We actually don't know. Odds are pretty high. We'll learn that there won't be any effects. Even higher than that, probably next likely would be that there would be some ill effects, some loss of function sorts of things. But it could totally give them new characteristics that make them better, make them more fit. In whatever nature that is. It could be one specific protein that they will have that will give them an evolutionary edge, a survival edge, an attractiveness and reproduction edge, an intelligence edge. Really, it only matters as long as they can pass those genes on down to the next generation. So linear DNA, like what we have, eukaryotes have strands of DNA, chromosomes, right? DNA polymerase actually cannot get all the way to the end of the 5' ends of daughter strands. It's a limitation of being required to have a piece of RNA there as a primer to build onto. And since we're not necessarily going to get a primer all the way up at the very, very end of a molecule to build onto, there's always going to be a little bit of DNA that is left off at the end before any primers can attach that does not get copied. This means that with every replication, our DNA gets shorter and shorter. And there's some conflicting theories, but this is theorized to be one reason why we cannot live forever, is because after a certain number of divisions, your chromosomes just get shorter and shorter and shorter and shorter and shorter, and eventually the chromosomes get short to the point where your genes start getting literally eaten with every replication. This problem is not an issue, by the way, for prokaryotes. They have circular DNA. There's no end on them. DNA polymerase gets started, it comes all the way around the loop. There is no 5' end that needs a primer that doesn't get exactly matched. So that is a problem for being a eukaryote. We'll learn what some of the advantages are for being a eukaryote later. It's also been shown that the reason that you can still have offspring and they can still have offspring and their DNA is not shorter than yours. There are some nucleotide sequences that get added on to your DNA only for your germline cells. So only for the stuff that's getting built to build your next generation. There are telomeres, and these telomeres are regions of basically just a whole bunch of extra DNA sequence. The extra DNA sequence is like a buffer for that being eaten down. And in your cells, the telomeres are not active. You don't have growing telomeres. However, it has been shown that there are some cancers where the telomerase has been activated, because you would think, oh, this is a nice thing. After a certain number of divisions, the cells can't divide anymore and they die off. And that would be an automatic defense against cancer. And that is one of our body's automatic defenses against cancer. If you have uncontrolled cell division, that's not supposed to happen. After several years of that uncontrolled cell division, those cancerous cells should die off. However, occasionally, those telomerase molecules or telomerase enzymes get turned on, and the gene regulating those adds more DNA to protect them. And this is, in some cancer cells, it has made them to be what's called immortal, meaning that we could take that cancer, we could grow it in a dish, and we could grow it for years and years and years, and it will continue growing forever. There was a woman in the 1950s whose name was Henrietta Lacks, and she had cervical cancer. She went in for an exam. They took a biopsy of her cells, which was commonplace at the time, and kept it for research at Johns Hopkins University, or Johns Hopkins Hospital. And they did research on this, and they found that she died a few years later from her cervical cancer, unfortunately. However, her cells are still around and happily living as cancer cells in labs all around the world, and you can purchase them for research. There are some huge bioethical problems with taking somebody else's cells and using them without their consent, but she was the first person to be found who had immortal cells, and that's because those telomeres continue adding on little bits of DNA to her cells. So it has been really useful for our research and a lot of what we know about genes and activity in human cells, and being able to do really good reproducible experiments has been thanks to Henrietta Lacks. So what would happen if telomerase did not function properly in a cell? Let's say, what would happen if telomerase wasn't turned on for your germ cells? Or what would happen in you if telomerase was turned on and it was turned on in overdrive? What would happen to the amount of DNA in you? Why does one strand of DNA lag behind the others? So why are these not copied at the same rates? And what would happen if a DNA repair polymerase ended up repairing things wrong and causing something that eventually led to a single amino acid substitution? Remember, DNA codes for proteins, and proteins built by amino acids. So it could be glycine, valine, cysteine, alanine. Instead of that sequence, one of those amino acids is swapped out for a threonine. What could happen there if that protein was built all the same except for a single amino acid was substituted? And what would be the potential implications of that? I guess I'll answer these. If telomerase did not function properly in a cell, like if it did not function properly in your germ lines, you would give birth to somebody who has a shortened life expectancy because eventually their DNA should get too short. If their telomerase did not function properly, by the time their offspring and the next offspring are born, their chromosomes have been reduced to being too short to be functional, and that would not be evolutionarily sustainable. So that would be a trait that would not continue on in the population. If you had telomerase functioning in overdrive or turned on in your cells, you might become immortal. We're not positive of other factors yet. You might live longer. You also might end up having too much DNA. So your cell copy rates could be decreased because the DNA quantity could your cell could spend its entire metabolic energy on just copying the DNA. Why does one DNA strand lag behind? It's because DNA can only be built in one direction. So one strand is made continuously, the other one has to be written backwards in segments called Okazaki fragments, named after the scientists who discovered them. And if we had a single amino acid substitution, if that amino acid is really similar in nature to the amino acid it replaced, it might do nothing. If that amino acid is different, it's going to cause a change in the protein's shape, which could cause all sorts of stuff. It could cause an increase or decrease in an enzyme activity. It could cause a protein to have a different job or a different color, or it could end up causing protein to be built way too small, way too big. It could cause a protein to lose its function and not do what it normally does anymore. So all of these mutations could result in genetic changes. Genetic changes can all potentially result in phenotype changes. They don't have to, but they might. Chromosomes, by the way, are a complex of both DNA and protein. This is review for you, but DNA is normally coiled into loops called chromatin. It's packed into a tight space. And those are our chromosomes, just giving you an idea of how a chromosome looks relative to the pieces of DNA we normally talk about. Heterochromatin is the really condensed, tightly bound up stuff. Euchromatin is a little bit more opened up. And this actually has to do with regulation of genes. Proteins bind to DNA and allow it to be either open, if it's what we call acetylated, then these genes are open and accessible for the machines of reading those genes. If they're called methylated, then that DNA is coiled up and those genes are not going to get used. We have a lot of our genes spend their time as being methylated, which is why some of our genes that we share with other organisms we don't necessarily use. So interesting to think. DNA builds proteins. Proteins have the job of also regulating the DNA. It's sort of a feedback. So your DNA can, through use, become methylated or acetylated throughout your life. So there is some evidence of genes being accessed and activated in the past, influencing future phenotypes. So what's the difference between heterochromatin and euchromatin? And what form would we expect to see in your DNA during anaphase, or really during any part of mitosis? What form would we expect to see a lot of your DNA in during interphase? And during interphase, the genes that are used or expressed, would they be in the heterochromatin or euchromatin form? Is DNA the only thing that determines your phenotype? Or do proteins regulate the DNA and how it is expressed? And I'm not going to have you do the last one. We don't need it for AP. So nucleic acid hybridization is making copies of nucleic acids and mixing them together. So we're gonna look at some of the biotechnology stuff that's here. Bacteria have. We already mentioned that bacteria have viruses that attack them. Bacteria also make use of plasmids. So they've got one big chromosome. They also have small loops of circular DNA that are called plasmids that they can easily exchange with one another. Bacteria do have sex, not in the exciting animal and plant sex sorts of forms, but they can, in certain situations, exchange DNA with one another. Usually when they're under stress, plasmids can be exchanged through some cytoplasmic extensions of bacteria. So these are ways that they are able to move around DNA between one another and share things like the gate keys into getting into our cells, as we saw at the very, very beginning of these slides, the mice that got sick from non-pathogenic bacteria getting a plasmid, probably from some dead bacteria. Remember, the DNA was still there even though the bacteria was dead. Bacteria also have some defense against viruses. They have restriction enzymes that are designed to cut some specific sequences called restriction sites. Go figure, these restriction sites are usually foreign to the particular DNA of that organism, but that bacterium, if it gets a virus and that virus injects some DNA, as we've seen viruses do, into that bacterium, if the bacterium has this enzyme that cuts up the DNA that it doesn't recognize, it's a good defense mechanism. We've used these restriction enzymes as some of our genetic engineering tools. So what do researchers do? First, we get a plasmid, which is a little loop of DNA, and we're going to take DNA from another source into it using some of these restriction enzymes to cut certain sites on these, and we'll make something called recombinant DNA. So here's the Cliff's Notes of the progress, and you guys don't get to do the lab this year, and I'm sorry. But we've got a bacterium with a plasmid. They isolate the plasmid by disrupting this bacterium, and we cut pieces of this plasmid and we cut pieces of this DNA from another organism using the same restriction enzyme. We're going to make some SNPs on it. We're going to allow those two to get together, and most of the time, this green plasmid will reheal itself, or it will anneal without getting any of this pink gene in. Or this pink gene will just never find its way in. But occasionally, we will be able to build this plasmid that has both the DNA from the original bacterium and this small piece of DNA that we want. This together works as a vector or a means of getting DNA into other stuff. We get this plasmid into a bacterial cell. We put it under a stressful situation, essentially, to allow it to accept this. Usually in the lab, we would put these on ice, then we would put them in hot water. then we would put them back in ice again. It's going to make their membranes do some weird stuff that can't keep up with the sudden temperature change. Some of those plasmids will get in, and as long as this piece of DNA is inside this recombinant bacterium now, it will follow the instructions that are on that DNA, and we have cloned a gene. What sorts of things might we want to do? Well, in class, the one that we were supposed to do this spring was take some DNA from jellyfish that is fluorescent. The DNA is not fluorescent. A gene for making fluorescent protein, and we put it into bacteria, and then that bacteria grows a fluorescent protein, and eventually we can get it to glow in the dark with UV light. Making things fluorescent is just a proof of concept, whereas we might have more interest on doing some other stuff. So if we had a gene that was in one organism that caused resistance to pests in plants, if you are wearing clothes today, and if you ate a meal in the past 48 hours, you consumed and are wearing some of the result of biotechnologically enhanced foods and crops. Your cotton that's grown in the U.S., 80% of it has been enhanced with these genes to prevent them from getting eaten alive by insects. Your foods in the U.S., something like 80 or 90% of them have been enhanced to give them genes making them more resistant to drought and insects. So it's something that's been able to give us a greater quality of life and a greater quantity of life. If we didn't have any of those, you should try growing some plants in your garden that are not genetically modified and see how they do in comparison to the genetically modified ones. You'll find that you end up having to use a lot more pesticides and poisonous sprays on your plants if you don't have these genes in them. So there is a definitive trade-off. You could not use the pesticides and not use genetic modification and let half the planet starve because we don't have enough food. That is a choice that your generation is going to have to make about what we do. We can also do things like build artificial molecules, like build medicines, build human growth hormone in bacteria, and then use it in humans. Build drugs that are useful and really expensive to produce. Otherwise, we can just enslave bacteria to make the drugs for us if we just give them the instructions and force them to do it. So it's useful. You need to know what is involved and be able to make some of your own decisions with it. So cloning is making multiple copies of a gene, and it's also talked about as being cloning if we take a gene and put it into another organism so that it's going to make copies for us. Amplification is how many copies we end up making. We're going to take a look at how the restriction enzymes work. Remember, these are defensive enzymes that end up cutting apart foreign DNA that goes into bacteria, but we have stolen these tools, and we haven't made any restriction enzymes of our own. We're just taking them out of nature and then using them for biotechnology. So let's take a look at what restriction enzymes look like.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT.
All right, so you can see right away why this might be useful for cutting out a sequence. If we've got this and then down the road we've got a useful gene that we want, this can be used to cut out a sequence of that gene. If we've got this sequence, which AATT is small enough that you can find it all over the place in your DNA, cut out some genes using this, And then these are called sticky ends because if we cut this piece off of our gene of interest and then we cut another piece in our plasmid, it's going to have the same end on it. It's going to be relatively likely that if we mix the two together, some of the DNA from our gene of interest and some of the DNA from our plasmid will happen to find one another and stick together. So that tool is used for us to be able to make recombinant DNA. Come on, baby. So here's one such example. We take a bacterial plasmid that exists. We cut this restriction site using a restriction enzyme, and we have our other gene of interest that's been cut using the same restriction enzyme from another organism. Most of the time, these two are going to stick right back together and make a regular old plasmid. Occasionally, though, we'll get lucky and we'll get this combination where we've got our plasmid, our gene of interest has been inserted, and we go back into the plasmid, and we have a recombinant molecule that is all together. Sorry, I forgot I made some bigger versions of this. So this is cut right along these lines in here, and gives us our recombinant plasmid. This recombinant plasmid is really expensive to make because you can see most of the time it's not going to work. However, once we get it to work, we can make more of it. We can put it into an organism and ask that organism to make more. Skip. Gel electrophoresis is another thing that we're not going to get to do in lab. You guys remember doing chromatography, right? We took a little piece of paper, we rubbed some spinach and maybe some other leaves on it. We put it in some solvent, and we allowed the solvent to move those proteins, but the colors up the colored pigments up the paper. And they moved different rates because they might have had different attraction to the paper, or different size, or different solubility in the ether or acetone that we used. Well, gel electrophoresis uses a very similar sort of technique. You chop up some DNA using restriction enzymes, and then those restriction enzymes cut up the DNA based on where some specific sites are, and they cut it into different lengths. You take those pieces of DNA, you put them into this guy, which is a gel. It looks almost like gelatin, but it's agar gel, and it has some little slots that are cut down into it. You dump these into the little wells in the gel, and then you apply an electrical current across this whole system. You put it in some basically salt water to allow electricity to conduct through it. You have to conduct your electricity the correct direction. DNA has a negative charge because of the phosphate groups on it. I had a couple of groups in my class last year connect their system backwards, and the DNA went bloop, fell out the back end of the gel. But DNA is pulled by the electrical field that's applied to this toward the positive end. And what we end up seeing in these is that the different pieces of DNA move at different rates depending on how large they are. So the smaller fragments move a greater distance from the well. The larger fragments stay as closer distance from the well. This was the original means of DNA fingerprinting. Now we can do actual gene sequencing and have people sequence in a couple of hours. But this was the original DNA test that was done to say, oh, we've got these particular known ones in our killer, and we'd look at our different samples from different suspects and find which one matches. PCR, polymerase chain reaction, is made for amplification of a specific gene. And this was brought about in the 1990s by a scientist who found inside thermophilic bacteria in hot springs, there is a polymerase that is an enzyme that is able to work at temperatures as high as boiling point. So PCR does cycling, where a piece of DNA, big G DNA, is put into a chamber with some specific primers and this enzyme. It's heated up to almost boiling point. They get it to like 95 Celsius. that's enough to denature the DNA or dissolve its hydrogen bonds. The DNA falls apart. The primers happen to attach. Remember, these are little sticks of RNA. They happen to attach as the DNA is cooled back down from near the boiling point. And then the polymerase that's able to operate at these high temperatures builds new nucleotides, and it'll build all the way down to the end. As this gets copied, this is going to cycle multiple times. Because these primers exist in certain regions, we're only going to get, by the end of this, we're going to end up with some DNA that has, some lopsided ends on it that are hanging off. However, by a few cycles in, we're going to get mostly just these pieces that are double-stranded for the target region. We're going to keep cycling this a few more. Each cycle on here might take about 30 minutes. So you might think, wow, cycle 3, that's an hour and a half in. Well, if we take a look here, after 30 cycles of this, we've had billions. So you let this go for a few hours, and you've got all the copies of your target gene that you need. And this is useful for giving us enough copies of something to actually be able to see it. So the pieces of DNA that were looked at in the gel electrophoresis, you have to amplify those pieces of DNA before you can use them. The pieces of DNA that we saw being inserted into plasmids, you'd have to amplify them because we want lots of copies so that the likelihood is high of us being able to transform them into another organism. Now I'm going to skip here. Recombinant DNA, we've seen that. Finally, we can do some DNA sequencing with those large quantities of DNA. In the labs that you guys will visit, and some of you will end up working at in college if you work with your professors in any of your biology labs, you will have tabletop, about the size of a laser printer, DNA sequencing machines that can take an organism and sequence it, get its entire DNA sequence in a few hours. So pretty exciting as what has gone on for these. The stuff that can be done within these was originally done, the first big project was done with the Human Genome Project in the 1990s, was done to sequence the human genome, all of the genes that are present within us. And it took about 20 years and thousands of researchers working in labs all over the world doing painstaking techniques of gradually adding nucleotides to DNA and figuring out what's going on with them. These next generation sequencing machines do something called flow cytometry, where they will feed DNA through a small port. They hit it essentially with stuff like lasers, and those lasers feed back based on the different nucleotides, which nucleotide it is. And if you get a flowgram like this, a former on this one would indicate that there are 4 guanines in a row, or in this case, there are 2 adenines right in a row. So this would read like T, C, A, G, etc. And then when you get to this point, it would be A, G, G, as it goes. So we're even seeing things that are probably going to be done in the next couple of years, where we have DNA fed through stuff like a nanopore, and then we're going to be able to read its signature even faster. So you may get to the point within your lives where we can sequence somebody, taking a tiny bit of their saliva, take it to a lab, have their sequence in just a couple of minutes, which is amazing to think about. So biotechnology is always looking for new tools, and the big dog in the past decade has been CRISPR-Cas9 or CRISPR-Cas9. So Cas9 is a tool for cutting DNA. It's like a restriction enzyme. It's a nuclease that cuts stuff, but it is guided by a piece of guide RNA. essentially you can give it instructions to cut where you want it to cut. We don't have to look for a specific sequence that's found in restriction enzymes and bacteria. We have the capability of inserting instructions into it. So the first thing people did with this was be able to disable genes, and this is the easiest way for us to determine what a gene does. We've got a gene sequence. I don't know what it does. We don't know if it makes a protein. Let's turn it off. You break the gene, see what happens. In the past few years, we've also been working with this to repair genes. So think about somebody who has a debilitating disease that's genetic. This system can edit their DNA. So let's take a look at what goes on with there. The Cas9 nuclease protein, it's got a cut site where it can cut DNA, and the guide RNA can be any piece of RNA that we come up with, or any piece of RNA that we build to put with this. You put the two of these together, and this will seek and destroy whatever target sites you want. So it is totally possible to do some gene editing of a living organism at this point. Now, you might look at this as being great. You know, my aunt has cystic fibrosis, and I want to use this to cut out the bad sections of DNA in some of her lung tissue and replace them with others. That's cool. I think that's a novel thing. We could use this to cure cancers and things, especially if we can sequence them quickly. We can build some RNA and solve their cancer. Where this gets scary is I could take an embryo of mine and my wife's. We could sequence that embryo, just a few cells, and I could find, well, I really want my kid to have good vision, better teeth, and be smarter and more attractive than the rest of their contemporaries. And we could use this same system to incorporate some other genes into them. And that's where we got to figure out, how far do we go before we say it's too far, and where do we draw the line? Clearly, science has the tools for these. Clearly, it is being used in some cases for really good stuff. But yeah, you guys are going to get to decide on how far and where that goes. Let's see. I don't know why I have that in there. Oh, okay. Last question on here. So we've got this is some of the original data from Erwin Chargaff. You can see this. 1952 is the published date on this one. So he looked at stuff from all over the place. We can see that Chargaff's rules indicate that adenine is always roughly equal to, and cytosine and guanine are always roughly equal. So my last question for you here is for you to take the ox with 29% adenine and fill in the rest. What's the guanine, what's the cytosine, and what's the thymine within the ox? You should be able to do it.